Good morning, everyone. Thanks to the organizers for the opportunity to tell you about our work. In today's talk, I want to give you an example of how systematic studies of protein localizations and protein-protein interactions have transformed our understanding of an organelle and our ability to engineer it. In our case, the organelle is the pyrenoid, a structure found in the chloroplasts of nearly all eukaryotic algae, and the pyrenoid plays a major role in the global carbon cycle. In the background of this slide, you can see a pyrenoid in a beautiful electron micrograph from Robin Roth and Ursula Goodenough. The pyrenoid is essentially a machine for force-feeding the cell with CO2. It consists of three subcompartments. There's a matrix colored in green, which contains the enzyme rubisco, which fixes CO2 into sugar. In fact, all of these little blobs are rubisco holoenzymes. The matrix is traversed by membrane tubules, which we think feed the rubisco with concentrated CO2, which makes the rubisco run faster and thus enhances the algae's growth. And the matrix is surrounded by a starch sheath, which we think serves as a barrier to slow CO2 from escaping the pyrenoid. A lot of solid science suggests that if we could engineer a pyrenoid into land plants, this would increase the yields of major crops like rice and wheat. But until very recently, the pyrenoid has remained almost completely uncharacterized at a molecular level. So if we dream of engineering a pyrenoid into land plants, we need to first address a number of basic questions like, what are the protein components and how do they work together to assemble this structure and make it function? To address these questions, my lab used a strategy where we systematically determined the localizations of candidate proteins. Luke Mackinder, at the time a postdoc in my lab, who is now running his own lab in the UK, started with 146 candidate proteins and developed a pipeline for tagging each of these proteins with the fluorescent protein Venus and using confocal imaging to determine each protein's subcellular localization. And we were thrilled to find that some of the candidate proteins localized to the pyrenoid. And this was intriguing because for 25 years, the pyrenoid has been thought to be primarily composed of two proteins, rubisco and rubisco activase, which is essentially a chaperone for rubisco. In these confocal microscopy images, the fluorescently tagged protein is shown in green, and the surrounding chloroplast is shown in magenta, visualized through chlorophyll autofluorescence. You can see that each of these fluorescently tagged proteins localized to a bright punctus in a hole in the chlorophyll autofluorescence, which corresponds to the pyrenoid matrix. And so you can imagine that we were thrilled when we started to see other proteins that also localized to the pyrenoid, like this protein, which also localized to the pyrenoid matrix, or other proteins that showed new, previously undescribed patterns of localization to the pyrenoid, like this protein, which we think localizes to the starch plates, or this protein, which appears to form a mesh around the pyrenoid, and we hypothesize fills the gaps between the starch plates. And my favorite, this protein, which shows a beautiful star-like pattern that traverses the pyrenoid matrix, and we think is sitting on these pyrenoid tubules. We then went on to perform immunoprecipitations and mass spectrometry on these and other pyrenoid localized proteins. And together we identified a total of over 90 high confidence pyrenoid proteins. In fact, the project has been so successful that we're now expanding it to the scale of thousands of proteins that are associated with the chloroplast. This spatially resolved protein interactome of the pyrenoid really opened the doors to its molecular characterization, and I want to summarize some of our discoveries that this study enabled. I just told you that the pyrenoid enhances CO2 fixation, and how we used a systematic localization and protein tagging and interaction study to gain insights into the anatomy of the pyrenoid. This protein atlas of the pyrenoid enabled a broad range of discoveries, including the discovery that the pyrenoid matrix is not a crystalline solid, but rather behaves as a phase-separated liquid-like organelle, which mixes internally. And knowledge of this protein localizations and protein-protein interactions also enabled us to rapidly advance the molecular understanding of the pyrenoid structure, including our discovery of this red linker protein, which holds together the blue rubisco to form the pyrenoid matrix, as well as our unpublished discovery that many pyrenoid proteins share a common protein motif that binds to rubisco and allows the three subcompartments of the pyrenoid to be glued together. Finally, our pyrenoid atlas has provided us with a list of parts that, with our collaborators, we are engineering into land plants in early efforts to reconstitute a pyrenoid into crops that we hope will increase yields of wheat and rice. So zooming out, I hope that these advances illustrate just how powerful systematic characterizations of protein localizations and protein-protein interactions can be in the study of organelles, and how such data would be incredibly useful on a whole plant level. Finally, I just want to thank the members of my lab, our collaborators, and our funding sources. Thank you for listening. I look forward to your questions.